Guys. So this is my presentation on the WI38 cell line. First, let's go over the fundamental biological concepts, which consists of the basics of cell division and mitosis. So, cell division. It's the biological process by which one parent cell divides to form two or more daughter cells. And in this process, we go through another process called mitosis, which is the process in which a cell replicates its chromosomes by segregating the chromosomes to produce two identical nuclei in preparation for cell division. Now let's go over the phases of cell division. Stage 1. This is the phase where cells prepare for division. There's the G1 phase, where cells grow in size and organelles duplicated. There's the S phase, where DNA replicates. Then we have the G2 phase, where cells continue growing and the DNA checks for errors. Next, let's go to stage 2. We have prophase. This is where the chromatin gets condensed and the nuclear envelope disappears, and spindle fibers begin to form. Stage 3, prometaphase. Chromosomes start finding and attaching to the spindle fibers. Stage 4, metaphase. The chromosomes align in the middle of the cell, the metaphase plate. Stage 5, anaphase. The centromeres split, the sister chromatids separate onto opposite sides of the cell. And then stage 6, telophase. This is where the chromosomes arrive at the opposite poles, and then the chromosomes decondense and the nuclear envelope reforms. And last stage is cytokinesis. This is where the ring of protein with actin and myosin pinch the cells, creating what we know as the cleavage furrow until the cells split. This is a demonstration of the cell cycle, a more visual demonstration to show where each stage is occurring. Now let's go over some history. The WI38 cell line and history. In the 1960s, there was a serious issue with the polio vaccine. At the time, it was difficult to find cells that scientists could grow in the laboratory for producing drugs and vaccines. The Hayflick limit told us that if you grew cells in a petri dish, sooner or later they would stop dividing and die. Of course, vaccines and drugs already existed for centuries before the 1960s. So how did scientists get around this? Scientists got their cell supplies with fresh ones from new animals or using cancer cells, since cancer cells just divide and divide rapidly. This created a big issue with the polio vaccine, though, as the cells were taken from monkey kidneys. And unfortunately, some of these cells were infected with the simian virus and is thought to have infected up to at least 30 million Americans. It suddenly became necessary to find an alternative supply of cells. So, by sourcing fibroblasts of an electively aborted fetus, Dr. Leonard Hayflick, who is this guy in the right above me, he incubated some of the cell tissues and added an enzyme to break down proteins that held the cell together and the growth medium that cells needed to just divide, as well as a growth medium solution which contained the nutrients they needed to divide. After a few days, he was left with a continuous sheet of cells, and one of those cells became the beginning of the WI38 cell line. But why are these cells so special? Well, Hayflick discovered that if you siphon off some of these cells each time they divide and freeze them, a single source can theoretically provide almost an unlimited supply. And this is due to the fact that most of the cells have 50 divisions left, meaning if you freeze them before their 50 divisions are done, you can, after each cell finishes their cycle of divisions, you can unfreeze them and basically you have like an unlimited sample, and then they'll keep dividing. And now, let's go to the medical impact. So let's talk about some contributions that this WI38 cell line has had to medicine. The WI38 cell lines have saved millions of lives by providing a safe and reliable platform for vaccine development. Fetuses especially are thought to be the cleanest possible source of cells, since they are less likely to have picked up any viruses from the outside world, which might contaminate vaccines or confound the results of experiments. Back in 2017, Hayflick was asked to quantify exactly how many lives the cells had spared until that point. And by comparing the global prevalence of certain infectious diseases in the 1960s, when the cell line was discovered, with the prevalence of infectious diseases then, He calculated that the vaccines made with the WI38 may have prevented around 4.5 billion infections in total. The cells are likely to have spared 10.3 million lives. There's also the fact that like like using these cells were much less costly than getting animal source cells since they required excessive monitoring to both maintain the health of the animals and ensure the viability of the research. Since the cells had divided relatively a few times, like I mentioned before, when they were collected, they could be grown for longer than before they reached the Hayflick limit, like I mentioned previously, which was the main problem we found. Most MI38 cells have 50 divisions left, which each take 24 hours to complete, so they can be grown continuously for 50 days before you need to restart again. And here are some of the types of vaccines that the WI38 cell line has helped create. We have varicella, rubella, hepatitis A, rabies vaccine, but we don't have any specific treatments that are non-vaccine related, but here are some images of the vaccines. And these are pretty like known these are pretty known infections. Okay, now let's go over some controversies. Some people that are morally opposed to abortion criticized the cell line for its origins from being an aborted fetus. Another controversy was the severe lack of consent during this experiment. The woman whose fetus the cells came from did not consent to its use. In fact, she didn't even know about it until years later when she was contacted by someone from an institute who was hoping for a more detailed medical history. With the advent of affordable genetic sequencing, human cell lines contain human DNA, and WI38 will share more than 50% of its DNA with the fetus's mother. In this light, a lot of people consider that this situation is potentially representing a privacy risk. And this image shows the Wistar Institute in Philadelphia, where the WI38 cell line was first created by Hayflick along with his um, colleague Paul. Now let's go to my stance. So, in my personal opinion, I believe that consent is necessary for this, and so with my personal opinions on the ethics of the WI38 cell line is that if the mother of the fetus was able to give fully informed consent and the privacy implications of agreeing to her fetus's cells, then use the cell line. Because then I think personally, I don't see a problem with the WI38 cell line. But the problem is there was no consent given. So that is questioning some of my morals. I also think that it's important to see the other side and ask what are the ethical implications of not using the cell line. Had we not discovered this cell line, millions of lives would have been either lost or severely impaired from the diseases and not having the WI38 vaccines to treat them. And so it seems like to me it was kind of necessary for the greater good. So it's kind of like a balance scale because there's no there's like there's wrongs and rights to the situation. Let's go over some policies and regulations that were created after the creation of the cell line. Due to the mother of the fetus not knowing her fetus was used in biomedical research, the controversies around the WI38 cell line and other similar cases helped push scientists, lawmakers, and ethicists to agree that people must be told what their tissues and cells are used for, and people must agree before researchers can use them. There have been many situations like this. This problem of lack of consent has been reoccurring as there's a similar situation that occurred in the case of Henrietta Lacks and the HeLa cells, where Henrietta was not asked prior to her death if her cells could be collected. So in both of these experiments, both women were unaware of the usages of these cells, and this raised severe ethical concerns, especially in the realm of public policy and consent to human subjects in experiments. So while the WI38 cell line didn't directly create institutional review boards, it played an important role in highlighting the need for them, especially as part of a larger pattern of ethical concerns in biomedical research during the mid 20th century, like I just mentioned before. And so, as a result of these concerns, the WI 38 being one of them, we saw the formation of IRBs or institutional review boards, which now oversee research involving human subjects to make sure that they all meet ethical standards. Um, there was also a major change in the U.S. in 2011 when the U.S. Office of Human Research Protection provisionally proposed the first major revamp of rules to govern human subjects in experiment. And this meant that written consent was required for the use of any leftover tissue after surgeries or procedures. And it was crucial, and this had to be maintained in order to maintain trust within the public. Now, my personal reflection. For me personally, the WR38 cell line has definitely had an impact on my life, even if it wasn't as drastic. For me and everyone I know, the WI38 cell line produces vaccines that have been incorporated into our routine immunizations that prevent me and my community from receiving these illnesses. And I'm able to live my life without the fear of contracting any of these life-threatening diseases due to the single cell line. My personal impact. The personal impact is that ethically, I believe that the origin of these cells was sourced wrong, and if it was just acquired with informed consent of the mother, the cells use would be much more justified, especially when the results can save generations. The whole origin is like a double-edged sword since the sourcing of the cells was unethical, but the impact it had was monumental. Luckily, the WI38 story sparked change for public policy, so that was one good bright side to it. So hopefully ethical issues like this in the future in these great scientific endeavors doesn't occur again. And this is my work sentence. Thank you.